So today I want to tell you a little bit of maybe controversial reasoning on oxygen, this wonderful molecule. It is a very hot topic recently, and it is very interesting to see that we are learning so much again from this molecule that seem to be something that we should know all about, and instead we are learning again. And I think it's in 2005 when this paper came out from the National Geographic, it was published on Science. This group led by Falkowski showed that about 5 billion years ago oxygen was already present, and they studied by they found it through fossils, through glaciers analysis in the Arctic and Antarctic. But the reality is that the real surge of oxygen on Earth was 2.5 billion years ago. I think it is quite impossible even to realize the timing, but reality is that very, very short, not really long time ago, only about, as you can see here, I don't have really the pointer, but about 50 million years ago is when the origin of placental mammals came on Earth, and that coincides with a surge of oxygen in the atmosphere, going from about 10%, as you see the red line, from 10% to about 20% in the atmosphere. And that makes sense, meaning that they needed placental mammals, they need oxygen to go from blood through the placenta and therefore deliver to the babies. But so going really to the end user of oxygen is really the mitochondria. And the mitochondria is this wonderful small organ, maybe a prokaryote, that was included in the eukaryotic cells. It's very nice because it has these two beautiful, one outer membrane and one inner membrane. And the inner membrane is really the focus of the attention where oxygen is used. What are these mitochondria? What do they do, these mitochondria? Their major function is to break down food, to produce cellular energy, and also to produce building blocks for growth. The inner membrane is really the place where the oxidation phosphorylation reactions happens, which is really it's a process in which electrode donors and electrode recipients they balance so to produce high energy bindings that are phosphorol binding to produce ATP. And these complexes, series of complexes, are complexes that we name complex 1, 2, 3, 4, that are transmembrane. Complex 1 is the acceptor of hydrogens, and then the hydrogens, while the oxygen is the donor of electrons, so then to produce these high energy bonds. The oxidation phosphorylation is really an evolutive process in which really, as you can see, it's really a trade-off between what is glycolysis and what is oxidation phosphorylation. And the reason why is that glycolysis is a very fast reaction but produce only little energy. At the same time, the oxidation phosphorylation in the mitochondria, it's a slow reaction that requires oxygen, and however it produces lots of energy, so about 30 ATPs. One is located in the cytosol and for the glycolysis, and one is occurs in the mitochondria. Examples of cells: if you are a very fast reacting cell, like a tumor cell, you want to express lots of glycolysis. If you instead are a neuron, you want to produce a constant energy, and you need to be very effective, so you can produce more with oxidation phosphorylation reaction. Now, going from the production to the consumption is really regulated by the consumption and the transport, which we know that it depends on cardiac output, the concentration of hemoglobin, and the saturation of oxygen. Now, I think the key to understand also the transport and delivery of oxygen is just going back to the oxygen dissociation curve that shows that really if you have 100 of a partial pressure in the blood, you really do not increase the transport of oxygen, just because you completely saturated the hemoglobin, which is the transporter of the oxygen to periphery, because the dissolved, which is the partial pressure, the last part of the equation, really do not transport that much in the dissolved blood, in the dissolved plasma. So what is important is to bind oxygen to hemoglobin. And as you can see, from 80, pretty much from 70 of partial pressure of oxygen, there is not an improvement in transport of oxygen. Instead, it goes really deep, the curve goes down when you go below. In the 50 range and below, you have a really decrease in the transport of oxygen. So, by definitions, we have a hyperoxemia that occurs when the partial pressure of intraalveolar oxygen exceeds normal breathing conditions, leading to hyperoxemia, which is defined by an increased arterial oxygen partial pressure, and you can see moderate hyperoxemia is between 100 and 300, and severe hyperoxemia is more than 300. Hypoxemia, this is just by definition, is when arterial oxygenation tension is below 80 in the normal range, but hypoxia is defined when there is a failure of oxygenation at the tissue level. So you can have hypoxemia in a patient or in a normal individual without having hypoxemia. That's the case, for example, of high altitude climbers, right? Those healthy individuals, climbers at very high altitude, they have hypoxemia, so low levels of oxygen in the blood, but they are not hypoxic. On the contrary, you can have an hypoxic patient, meaning that there is a tissue demand of oxygen in a patient with a normal hypoxemia, such as in a very severe anemic patient. Why oxygen can be toxic? So now I'm going to, the next 10 minutes I try to explain the possible injury of hyperoxemia and possible advantages of hypoxemia. So oxygen can be toxic at several levels. First one is that if we think about it from an evolution point of view, none of us have been exposed to a high level of oxygen ever. We have been exposed to hypoxia environment, or actually our cells and all any kind of living cell during evolution has been exposed to hypoxic environment, but never to hyperoxic environment, only in the hospitals. And the reason, and the thing is that basically we do not have defense mechanisms, if you will, to hyperoxemia. We didn't develop. So when we have lots of oxygen, our mitochondria, if the oxygen enters in the electron transport chain, reactive oxygen species, they can accumulate, and our defense mechanisms that are either enzymatic or non-enzymatic cannot be increased just because we don't have the possibility, the defense mechanism of increasing these enzymatic and non-enzymatic antioxidants. So potentially we are exposed to lipid peroxidation due to reactive oxygen species, protein oxidation, and DNA damage. Pulmonary toxicity is really try to divide into two levels. So the alteration in gas exchange is driven by the inhibition of hypoxic pulmonary vasoconstriction and by the absorption atelectasis that increases the intrapulmonary right to left shunt. But also there is a direct pulmonary toxicity, so-called Lorenzo-Smith effect, that consists in reactive oxygen species related to direct toxicity of alveolar capillary barrier, and this leads to lung passage, congestion, hemorrhagic pulmonary edema. This is obvious for whoever worked in a laboratory with mice. If you did, or colleagues of yours, if you expose a mouse to 100% of oxygen, eventually the mouse die for pulmonary hemorrhage. Clearly this is not the case in humans, meaning that we are much more tolerant to high levels of oxygen. However, studies are now accumulating saying that there are possible patients that are more prone to develop toxicity, especially in hyperbaric oxygen chambers. There are cardiac effects that are decreased cardiac output due to both a drop of heart rate and an increase in vascular resistance, impairment of tissue perfusion, especially in the cerebral and the coronaries. And this hyperoxia-induced vasoconstriction may result from a fall in nitric oxide bioavailability, with further potential contribution in a reactive oxygen species. Neurological effects, this is increase in cerebral excitotoxicity with hyperoxia after stroke and hyperoxic perfusion injury. These are some of the studies that I just wanted to list it that have came out in the past 10 years showing that high and the elevated levels of oxygen in the blood may be toxic at several levels. We know from decades that oxygen is very dangerous for in newborns for retinopathies, but more evidence is accumulated regarding kidney injury, neurological injury, and myocardial injuries in newborns. This has been publication to Pediatrics in 2006. There is this story I spelled incorrectly. The point number 2, it's a Staub, S-T-A-U-B. It's a circulation paper in 2015 from the Australian group. They showed in circulation that oxygen supplementation in the emergency room for patients with ST elevation myocardial infarction that are not hypoxic at admission. If you give oxygen to those patients, myocardial size at MRI 6 months later is increased. So if a patient arrives in the emergency room, do not give oxygen if the patient is not hypoxic. Third, increase in ICU mortality in sepsis. This is a paper that came out on JAMA in 2016. This group showed that there was an increased mortality when oxygen was given as a conventional oxygen therapy at 60 days compared to conservative oxygen therapy, where oxygen was kept, oxygen was kept between 60 and 100 PaO2. There is also an increase in mortality after cardiac arrest. If you give a high level of oxygen, and there is also an increased mortality after stroke if a PaO2 is kept more than 300. This is actually just came out like a week ago, so the last number of the New England Journal of Medicine, showing from the Swedish group in about 6,600 patients, and what they did was to give oxygen to patients in suspected acute myocardial infarction, and patients that were not hypoxic or ambient air. And the patients that had ambient air, there were a group of patients also that they became actually hypoxic. However, both in both hypoxia and non-hypoxic group, there was no difference after 1 year in terms of mortality. So they didn't give any benefit. Oxygen didn't give any benefits in this case, but they also didn't give any. It didn't hurt. So in conclusion, I think this is actually a very interesting sentence from Paracelsus. It says, "Omnia venenum sunt, nec sine veneno quicquam exist. Dosis sola fecit, ut venum non fit." Which is, in a really summary, I would say that if there is no need to give oxygen, we don't need to overdose our patients with oxygen. Now I'm going to switch gears and a little bit talking about the possible benefit of hypoxia, so it's even more controversial, so I'm happy to discuss with questions. All of us we had a long time of hypoxemia, but we were just doing fine, and we didn't have any problem. In fact, we were very happy for 9 months when we were at this level, and our level of oxygen in the blood is about 20 millimeters of mercury, 20-30 millimeters of mercury, and we didn't have any issue. In fact, we were growing fast, our cells were multiplying quickly, so in a sense we have a system in our cells that can turn on and live a great life in hypoxemic condition. More recently, sports activities and sport medicine is going from what is called live high, train low, or now live low and train high, which means that there are more and more places like this, where there is a big company in New York that makes this hypoxico. You can visit also on the web, hypoxico.com, and then they are making any kind of environments for training you at a low level of oxygen to improve your endurance. So you have your hypoxic lap swim at 18, 15 of fraction of inspiratory. You can train it inside here, or you can even do any kind of activities and train in hypoxia. But there is also many people that live at very high altitude. If you think about at El Alto in Bolivia, there are 1 million people. It's like to take all Krasnoyarsk and going up to 4,100 meters. The level of oxygen in the blood, in the arterial blood, is 50. It's 1 million people that they are very happy. They have normal lives, they study hard, they use their brain, they do jogging, they do anything activity, they deliver baby, and their oxygenation in the blood is 50. They have pneumonia and they go to the hospital. They don't have much reserves, but this is very interesting that there is a population that at very high altitude that they live just a normal, regular life. If you have time, read this beautiful review from John West that came out on the New England this year. It's a very nice read, and it's fantastic. It's very well written, and a wonderful review on the chronic hypoxemia response. But why can we live at very low levels of oxygen? The reason is that the mitochondria do not need a very high levels of oxygen. In fact, the levels of partial POM, just partial pressure of oxygen in the mitochondria, is very low. It's about 5, 6, 7, 8 millimeter of mercury. And therefore, like in a baby, like in a fetus, 20 millimeter of mercury is all what you need. There is a very nice drop in oxygen. So it's very, let's say that it's very conservative to have 150 in the air in order to arrive to a very low level of oxygen in the mitochondria. But I think that, the reason is, so how can we, in a sense, respond to hypoxia? And what happens when we respond to hypoxia? The HIF factor, which is hypoxia-induced factors, is produced, and all what it needs to do is to go through the cytosol and enters the nuclei, binds HIF beta factor, and what they want to do is to go to the nucleus and to produce all these factors that are the hypoxia-induced genes. And what they do, really, they turn on glycolysis and many other they do also many other activation of many other genes we will talk about. But the main idea is that in absence of oxygen, HIF alpha, what all it wants to do is to go to the nuclei and produce these inducible genes. When oxygen is around, HIF factor is destroyed by ubiquitin and and the others ubiquitin that are attached to the proteasome and by hydrolysis is destroyed. So HIF is produced and destroyed, produced and destroyed, produced and destroyed continuously as long as the oxygen is around. When oxygen goes down, HIF if alpha is able now to execute the activity, which is entering the nuclei and activate the hypoxia-induced genes. This makes sense because in absence of oxygen, what we want to do is to turn off the oxidative phosphorylation reaction and activate the glycolysis. So first, as John West has described beautifully, it says glycolysis is activated, and then all these other pathways are activated, which is mitochondrial genes, angiogenesis, pulmonary circulation with the nitric oxide synthetase in order to decrease pulmonary vasoconstriction due to hypoxia, and then EPO, of course, because you want to have more mass of red cells, so erythropoiesis is increased, and then many other mechanisms. But keep in mind, I think the most important now is hypoxic metabolism inducing glycolysis. Now, the world, I think, has been kind of shaken by the idea that came out from this manuscript that came out on Science in 2005, that actually hypoxia can be used as a treatment. And this is now a step forward. So how can, I mean, until now we talk about hypoxia as an escape mechanism, so using a glycolysis as an escape mechanism. But in the case of mitochondrial dysfunction, and therefore when mitochondria are not working properly for one of the possible 4 or more mechanisms, but mostly due to the 4 complexes that we have in the mitochondria, or either for a defect of complex 1 or 2 or 3 or 4 or a variety of these ones, what you would like to do is to turn off the oxidative phosphorylation reaction to improve glycolysis, to decrease the injury due to the reactive oxygen species. And so this group of scientists, what they did was so they were thinking, how can we deactivate the mitochondria that is dysfunctioning and activate the glycolysis. So if you go back here, either you have an inhibition of the ubiquitin in presence of oxygen, or you have, okay, sorry, I will finish, or you have instead or as they said, they just exposed mice to very low levels of oxygen. And this is what they did, and what they showed in red, these are mice with a mutation in the complex 1, so they cannot phosphorylate, they cannot use oxygen, and so what happens is that by day 70 the mice are dead. And when you give hypoxia at 11%, those mice, 160 days, they all survive by activation of the glycolysis. If you instead give oxygen to these mice, they die by day 30. Now you say, yes, but most of our patients instead they come when they already have a disease, not when they are really this is a preventive mechanism by shifting directly to the glycolysis. Says, but our patients instead they come with already disease. So what they did was also to expose these mice after they have these already lesions, that is characteristics in the midbrain. After the late stage in these 4 mice, 1, 2, 3, 4, they have lesions in the midbrain that you can see where there is the arrows, and then exposed to hypoxia, and the brain lesions resolved by 4 weeks. And this is just was published on NAS. And I just want to conclude because sorry too long, but so the next step, what we want to do is how can we actually make sure that in healthy individuals we can activate now the hypoxic response. And so we did, in this study we just started, we just exposed some of our volunteers to very high levels, like just to bring it to about 4,000 meters. And what we wanted to, and we decreased the oxygenation from 16% to 11% over 5 days. And what we found was that besides this, so it goes skip, and we decreased the oxygen in the blood to 50, which is similar to what we found in mice, that was about 45. And what I think is important is that we were able to activate Hippo, which is one of the genes that are activated during hypoxia, and we were able to see a spike in reticular count over time, so a delay response, but it's in correlation with an increase in Hippo by day 5. In conclusion, I think that now what we are doing is a metabolomic and proteomic study of plasma urine to characterize the normal molecular response to hypoxia and return to normoxia. And together with other animal studies to different complexes of the mitochondria, we are studying where hypoxia can be targeted as a treatment. And we can consider hypoxia as a compassionate rescue for treatment for children with same defects of complex 1 mouse that we study with cerebral infiltration, using a combination between MRI and hydroxybutyrate levels that is really biomarkers to determine response. And with this I wanted to thank my mentor, which is Dr. Zepol, and Dr. Van Simuta, which is the mitochondriomics scientist where we are studying this mouse model. And here it's together with Luis Ignaro, which is the Nobel Prize for nitric oxide that came to visit our lab. Thank you.